eye dissection. So I'm just going to place a pig's eye on to the petri dish now. Now you'll see it's got a bit of liquid over it, and that's just the alcohol that we've used to preserve it. So that there is a pig's eye, and the first part of the dissection is to just get it open. So I'll do this by piercing it with a scalpel. So a scalpel is just an instrument they use to dissect with, and you can see it there. And it's quite a tough surface to get through, so that's why you have to use this sharp scalpel. See that little bit of liquid coming out as well. Vitreous, I think, is the name given to the kind of jelly liquid inside the eye, and that's just used to keep the pressure inside the eye correct, so that your eyes don't explode or contract too much. So this gel stays at the same density throughout a wide range of temperatures. So now just cutting around the eye, and we're cutting through the retina and the sclera here. So the black that you can start to see emerging inside is the retina, and the white that you do see on the outside of the eye is the sclera. So just cutting it open now. And there we go, so you can see the black inside, and also that white jelly type stuff, as I said, which is the vitreous. So the next thing I'm going to do now is just remove some of this jelly, which you can see there on the tweezers, which we call forceps in a lab environment. And that big piece there is the lens of the eye. A lot of people know about the lens of the eye, and that's what focuses. It's the same in the human eye as the pig's eye. So yeah, and that there is just more of the jelly and part of the retina, which is coming out now. There's also a bit of muscle in there. It's quite tough, fibrous stuff. Yeah, made out of proteins. So later on, you'll see a video coming up, and I'll do make some slides, microscope slides that is, of these different parts of the eye. So we've got some muscle, probably a cross section of the lens, part of the retina, iris, so on. We'll see the iris a bit later in the dissection. So there we go. See that's empty now, that bit of the eye. So just using the scalpel to carefully peel back this retina, which is the black coating on the inside of the eye that we can see. Unfortunately, it sticks quite well to the sclera, which means it is quite hard to peel off, just to show in the camera. So there we go. It's not really coming off as well as I would have hoped in this dissection, but there'll probably be another video soon enough where it does work. So now I'm just going to cut using the scissors again, and what we're going to try and do now is take out the cornea and the iris altogether. So the cornea is like the first layer on your eye, and it's a clear coating, and then the colour, which in this case is blue, is the iris. And you can see, if you look at your own eyes, or look at someone's, or look at your own in a mirror, the iris, and that's your eye colour, and the cornea, you can also see that. The iris also contracts and goes in, and all of that to help you bring in different amounts of light. So if you're looking in the dark, then the iris might open up quite a bit, like it letting in lots of light. but if you're in a very bright environment, snowy environment, let's say, and the iris closes up, letting in less light to help you focus on certain objects. Now, the black that you can see coming off now is the retina. Finally, it's peeled off. It's quite an awkward piece, and you can see part of the iris attached to that retina, which is that ring that you can see on there. So, yeah, so far we've got the lens, the retina, part of the sclera, we've got the liquid, the vitreous, and I think I'm just about to cut now to get the cornea, which, as I say, is that clear coating that you get on the eye, which you can see if you look at your own eyes or something. So there's quite a bit of work getting this off. The eye's made very tough, so it doesn't break, and there's many cells thick. But you do have to be careful with your eyes, because they don't recover once they're damaged. If you damage the nerves and the sensing cells at the back of your eye, they don't grow back. But stem cell research is proving that wrong recently. So that there, what you can see in the tweezers, the forceps, is the cornea, and a bit of the black on the outside is a bit of the retina. And that white there is the sclera. Now, what you can see in the middle at the back there, that's just that there, is part of the nerve which would send all of your eye signals down your spinal cord and to your brain, so that you can actually see, because your brain is the part that actually processes the signals that come from your eye. So your eye is a sense organ which contains sense cells, which are special light sensitive cells which are at the back of the eye. And what I'm peeling off now is a bit of muscle. So there's always muscle around the eye, which helps to fit it in to the eye socket. So it's not an easy dissection, because it's quite a small object to dissect, but hopefully you can see the different parts coming together now and see how they all do fit together in an eye. As I say, this is a pig's eye, but quite similar to a human eye. So it feels quite cold after being in the alcohol. So there we go, we can see the different parts of the eye there. That about concludes the pig's eye dissection. There will be more dissection videos coming up soon, but for now I'm just going to say thank you for watching my video on the pig's eye dissection.